אוקיי? נהדר. תודה רבה.
There's no one to really present the speaker and everything. So, please start.
Very good. Okay. Good morning, everyone. It's a real challenge to talk about RNA regulators in an ELSC seminar, but I'll do my best. And they do relate to neurotransmission, so there is a logistic explanation. So we are talking about acetylcholine, microRNAs that enwrap around messenger RNAs to block their function, transfer RNA fragments, which are our most recent favorite for research, and the general regulation of cholinergic genes by these small RNAs in men and women, mainly with mental disease or underaging and neurodegenerative disease. And I'd like to start that by telling you how I got introduced to transfer RNA fragments. So the man on top was my late supervisor, Uri Litauer, at the Weizmann Institute. The picture in the bottom is Francis Crick, who officially declared after the discovery of the human genome that 2% of the genome encodes for proteins and the rest is junk DNA. So I said that recently in a seminar and people said, no, impossible. It's not possible that a Nobel Prize laureate would say something like that. But he did. I was teaching students using this citation. So for a long time, we thought that 98% of our genome means nothing. Today we know that this is the book of instructions of how DNA can be used. So it's definitely very, very important. What about transfer RNA fragments? So again, my supervisor published a paper in Science saying that bacteriophage-induced transfer RNA gets degraded. You look at it today and you say, well, this is reminiscent of the CRISPR mechanism, but of course in 76 no one even thought about that. So there are transfer RNAs, they get degraded, and that was the only RNA that people could sequence those days because it was before Sanger sequencing was invented. So I had a PhD project. My PhD project was to take the bottom left quarter of tyrosyl tRNA and synthesize it using a bacterial enzyme called polynucleotide phosphorylase. This is a fun enzyme because it can either synthesize or degrade RNA chains according to salt concentration, and it had to be purified. So I dived into the cold room for 2 years. I purified the enzyme, we published it. I was very pleased with myself. And then all I had to do was to take the nucleotides, condense them chemically, purify them on chromatography, extract the product, purify it, add another nucleotide, and so on and so forth. Each step about 7% yield. And after 2 years, I had this paper, I had the project, I had 7 nucleotides out of the 24 that I was supposed to synthesize. This was very good. So I went to my mid-PhD committee, and there was a wise woman there, Sarah Fuchs from the Weizmann Institute, and she looks at me and she says, 7% yield each step. Did you ever think what will happen if you walk down the corridor and the tube falls? No, I didn't. So if there's any PhD student here, listen to your PhD committees. They're there for a reason. But she really shocked me. I went back to my bench and I thought, this enzyme can either synthesize or degrade. Why don't I use it to degrade RNA and study the functions of the degraded section instead of synthesizing? So to do that, I'd like to tell you, but I don't know how, okay. I'm not sure how to activate a movie. No. I don't know how to activate a movie with the clicker. Anyway, it doesn't matter, so we'll stop that. So okay.
יש למטה צריך להיות כזה...
כן, אני לא יודעת אבל איך מטה.
יש לך למטה החץ.
Okay, yeah, okay. No, whatever, doesn't matter. So I registered to an EMBO course, another good advice for PhD students. Go to EMBO courses, these are really precious, where John Gurdon was teaching. No, I did something wrong with the presentation. Oh, תודה רבה יונתן. Save me please. Okay. אני תמיד באה הנה עם מחשב, היום באתי בלי. זה לא עלה על דעתי. נהדר. Okay, so to cheer everyone up, I brought a movie that explains what are tRNAs for ELSC members. So tRNAs bring amino acids to build the protein chain. Each one of them is essential. If you delete or mutate tyrosyl tRNA, tyrosyl-related proteins will vanish. So each of them is really an essential part of a conveyor belt. And here you see what happens when one of those parts goes bad. I don't know who of you saw that movie, but it was really great. And it's a crazy picture. you need to keep that chain going. Okay. And it's a collection of circles that keeps protein chains being built. So what I learned in the John Gurdon EMBO course was how to inject RNA into eggs of frogs, Xenopus oocytes. By the way, this is the only scientific product that I know that arrives from South Africa. They collect them in marshes, and the label says wild females freshly caught. So it's a very useful research, essential thing. We learned how to inject RNA, and we injected globin RNA, which was the only RNA you could produce then, because erythrocytes make only globin. And we could show that the 3' tail of that RNA stabilizes messenger RNA. That became my PhD project, and everyone forgot about tyrosine tRNA. And when John Gurdon won the Nobel Prize in 2012, I was personally very happy. Then I went to New York and studied molecular biology. What I studied before was called biochemistry. I have a PhD in biochemistry because molecular biology didn't exist. But in New York, Jim Darnell was establishing the methodology for molecular biology. Then I got back to Israel and I decided to focus on a nervous system related topic, the control of acetylcholine functioning. So what we know about acetylcholine is that it controls functions both in the brain and in brain-to-body messages, and it can block inflammation in blood cells. So indeed, multi-tissue functions. And that, of course, relates to its capacity to regulate stress reactions, which are a very favorite topic these days. And those stress reactions would affect the brain, the blood, the cardiac activity, and inflammation. So we studied stress responses in Israelis. And that was 10 years ago, in 2014. But somehow we had the insight to add to a biochemical and clinical checkup of I think it was 18,000 people who come every year to an annual health checkup paid by the company that employs them, Jews, Christians, Muslims, all working individuals. We added a questionnaire including questions like, to what extent do you feel the impact of terror events on your daily life? I don't know how I thought about that then. Today it would be much more relevant, but that was 10 years ago. And then we measured cholinesterase activities in the blood, and we added a test of the pulse rate, which is so simple and so unsophisticated that many clinical trials just don't use it anymore. But the Chinese medicine knew to appreciate pulse changes as a medical reflector of a lot of activities. And what we found using machine learning was that among these 18,000 individuals, there were about 2,000 whose pulse changed by 1 unit every year. So these are the red dots. And 740 whose pulse changed by 1.5 pulse changes every year. And this was in 4 years. Now, this change is already significant. And if you read the clinical papers, it says these people are at risk for stroke, for heart attack, for death of all causes. So we compared that to the level of inflammatory proteins in their blood, and those whose pulse changed were at the top right corner, so their inflammation reactions were very high. And those were the people that answered 5 out of 5 on the question, to what extent do you fear the impact of terror on your daily life? So feelings, inflammation, and cardiac activity are all related. The changes were tremendous. An average pulse change at the age of adults would be about 60, and these people changed it by 8 to 12 beats on an annual basis, so that means they were at real risk. So this was covered, by the way, in the newspapers last week, so it is still very relevant. We sent the paper to PNAS. The postdoc, an ELSC fellow, Shani Shenhar, asked me, so what happens now? And I said, now you'll come to work every day, you'll get to the website of the journal, you'll find no change, and one day there will be a change, there will be reviewers' responses, and they will tell you do this and do this and do that to prove the concept, and then you'll know what you'll do in the next half a year. Except this is not what happened. After 2 months, the journal said the reviewers applaud the authors, because nobody looked at the impact of stress on entire populations, and we just improved since then. So maybe that would be the right time to look at stress reactions in the Israeli population. But I promised you to tell you about microRNAs. So if we look at DNA to RNA to protein as the pathway, then microRNAs wrap around target genes that are encoding proteins and prevent them from doing their functions. We have hundreds of microRNA genes in our genome, 1/3 of which is primate specific. So these are tiny genes. A microRNA is a very, very small sequence, so you can change them in 1 bit of a nucleotide modification, and that apparently still happens. So we are very much enriched with these traffic controllers that are microRNAs. They don't do anything, but they prevent others from doing something. I suppose you could call that a police of some sort. And that was the kernel of Shachar Barba's PhD here at ELSC. So how does this relate to acetylcholine? Does that control cholinergic activities? Probably it does. And then we wanted to find out how it relates to stroke. The increase of reach increases. Okay. So what do we know about ischemic stroke? Kasha Winek was a postdoc in the lab, actually the Shimon Peres ELSC postdoc. She's very proud of that. And she decided to look at stroke reactions, which are more prominent in, the risk for which is more prominent in men compared to women. And stroke patients, ischemic stroke patients, die not because of the stroke. They die because they get pneumonia. So what happens is the immune system is really down-regulated dramatically. And the idea was to look in the blood for their microRNA controllers. That was the aim. So how do you look for microRNAs? We ran small RNA sequencing, and we found collections of families of small sequences getting shorter and shorter, probably by nuclease degradation. And Kasha came to me and said, this is junk. And I looked at him and said, oh my God, that's the RNA I was supposed to synthesize as a PhD student. This is not junk. These are fragments of transfer RNA. And the issue was that part of them were complementary to cholinergic genes. So Sima Dubnov in the picture developed a PCR test to verify that these changes that we saw in the sequencing are for real. And then we decided to look for how many of them were targeting cholinergic genes compared to microRNAs. And we said if they are like microRNAs, they are small, maybe just like microRNAs, they can become therapeutic agents. So what do we know about transfer RNA fragments? Actually, over the last decade, it seems that these are as eminent as microRNAs, if not more. So one fragment of transfer RNA is essential for sperm maturation. Another one regulates ribosome biogenesis. The levels change under angiogenin, which is a nuclease that creates them, breaks tRNA into them. And we know that angiogenin levels go up under stroke. So then I said, okay, microRNAs are great. We studied them for 10 years. But suppose you are under an emergency situation, you need a small RNA controller. There is the option to transcribe and transport a microRNA. But that takes a long time. Transcription is slow. It's not an emergency tool. On the other hand, if you can use a hammer, break down tRNAs that exist in all cells, as we saw in the Charlie Chaplin movie, all you need is to cut them down, and you have a tool. You can start using that tool much quicker. So looking at that, we compared the levels of cholinergic microRNAs to cholinergic targeted transfer RNA fragments. These are the red dots. So the volcano plots show you that transfer RNA fragment levels go up dramatically. This is in the blood of stroke patients 2 days after the event, whereas microRNAs go down. And we called that changing of the guards, and it was before the coronation, so unrelated. And we said, okay, this is the reaction of the cholinergic system, and if microRNA fragments are doing something, then perhaps that may use another family of genes that no one thought about as inflammation controllers. So we looked at the cholinergic genes that are expressed in different blood cells. This is Sebastian Lobentanzer, who was then a PhD student from Frankfurt that was meeting me in any cholinergic meeting where I arrived in Germany and consulting with me. And then we started publishing together, and one day I got a letter from University of Frankfurt, your co-supervised PhD student, Sebastian Lobentanzer, which was an interesting surprise. Anyway, Sebastian could show that most of the cholinergic genes are expressed in CD14 lymphocytes, one cell type, and that the transfer RNA fragments and microRNAs target completely different transcription factors. So the impact on the cells would be very different if you use transfer RNA fragments compared to microRNAs. Again, we said this is a potential drug, and Sima wrote a book chapter. Sima is an ELSC student herself, and she could show that publications on transfer RNA fragments just started 10 years ago, but they are escalating very rapidly. They can be produced either from the nucleus or from the mitochondrial genome, which is important because we inherit mitochondria from our mothers. And each subfamily of them has a different function. Which brought us to the issue, where are the cholinergic targeted transfer RNA fragments coming from? And Dana Schulman found in a collaborative project with Jonathan last year that most of them are produced in mitochondria, which means that the capacity to control inflammation by transfer RNA fragments may be inherited from one's mother. And that was the case in the blood of both healthy individuals and Alzheimer's disease patients. So there are sex-specific declines in those transfer RNA fragments that target cholinergic genes, and we found that in the nucleus accumbens brain region of Alzheimer's patients compared to controls. Not only we found the fragments, but we found that they can target the messenger RNA in other brain regions. So the nucleus accumbens sends messages to cortical brain regions that apparently react to that by declining cholinergic genes. The cholinergic pathway is known, found by many others, to decline with age, and just using cholinergic targeted drugs after the age of 50 is considered a risk to one's cognitive capacities, including, for example, anti-allergy pills that may be sold without even medical advice. So Dana found that. She was a master's student under Jonathan and my supervision, came from the bioengineering program, and again she looked for the changes in nuclear originated and mitochondrial originated transfer RNA fragments, and look at the difference in men and women. Now, why is this new? Part of this being new is because women were not allowed by law to participate in clinical trials either in the United States or in Europe until 1977. So a lot of the clinical information on women patients of all diseases was obscure until quite late. And today it's still a real problem because most of the drugs we use today were developed by males, tested on male mice alone, examined in men in clinical trial participants alone, and half of humanity is actually using drugs that were developed for use by the other half. But if we look at the genes, we see that the females show changes both in nuclear originated and in mitochondrial originated transfer RNA fragments, and males don't. So this is a female specific feature. Why is that interesting? For decades we know that women who develop Alzheimer's disease deteriorate faster than men, especially at the cognitive level. But nobody knew what was the reason. Now here we find an element that blocks the cholinergic pathway, which is important for cognition. That is true both in males and in females. So could it be that these transfer RNA fragments are important for controlling the pace of decline? We looked for the clinical data of the women patients created in Rush University by David Bennett and colleagues, and what we found was that the change in transfer RNA fragments reflected not the pathology, but the cognition decline. So that seems to be a causally related change. And finally, we compared it to microRNAs, which are shown here below. And what you see is that transfer RNA fragments are being changed in females dramatically, but microRNAs are not. And if we combine the data, then we lose the difference. I get a lot of papers to review, especially cholinergic papers, and I always say, could you please separate the cohorts to males and females? And the common answer is, but then I would lose my statistical significance. I say, exactly, that's why you need to separate. And eventually I have to say to the editor, I'm sorry, I can't review this paper if it's not separating males from females. So this is a problem not only in Alzheimer's disease, but in many, many clinical studies. And indeed, you need a much bigger cohort to be able to separate males from females. But I don't see any other way where that could be done. That's Alzheimer's. What about stress reactions, which are very pertinent level? So I'd like to mention a study that we completed last year, which was done by Nadav Yayon, which was looking for the impact of stress, social stress responses in young mice. So we knew from the literature that in mouse colonies, adults tend to humiliate youngsters by plucking out one part of their whiskers, only one side. And those mice grow up to be depressed forever. We know that the whiskers are controlled by the barrel cortex, so on the contralateral hemisphere, so we knew where the neurons are. And the question was, how come? Why does this social stress persist for so long? And the barrel cortical neurons include cholinergic interneurons. How do they react to this mild stress? And Nadav, who is now an EMBO postdoc in Cambridge, UK, divided the cholinergic neurons in a labeled strain of mice into little boxes and analyzed each separately. And that enabled him to label the neurons and compare their structure within the cortex. I know that the cells look very densely arranged, but actually they are very sparse compared to other neuronal populations. And what Nadav could find is that the structure is changed forever. So if we talk about those children who were released from Gaza, they need to be watched after very carefully, because stress conferred on very young mammals may change the entire cholinergic network forever. So what Nadav found is that the branching pattern was changed, and that would change also the connection in the network. So how do we proceed from there? How different are short RNA profiles between the distinct cell types in the live human brain? So our lab collaborates with the Dopaldor at Shaarei Tzedek, and we have a Helsinki approval whenever he operates on a brain to remove metastasis of meningiomas or something like that. He's allowed to take tiny pieces of tissues, and he brings them to the lab, and Sima extracts RNA. She's now trying single cell sequencing on those tissues. And of course, the main issue here is, to what extent are we limited in studying post-mortem tissues? Would it be different if we looked at cell types extracted from the live brain? And what Sima is trying to find out is the profiles of small RNAs, because single cell sequencing does not yet allow to do that. So tRNA fragments and microRNAs in different cell types that are brought from IDO, and the methodology is actually the one we developed with Nadav in that stress study. So we separate the cells, we can purify neurons or other cell types by antibodies, by FACS sorting, and then we find out how many of them or each type exist in particular cell types. And what you see here is that both microglia and neurons include microRNAs, which was not a big surprise. The surprise was that neurons have many more transfer RNA fragments than microglia. So whatever we see there is really neuronal reaction, and this is valid for the live human brain. So Sima proceeded to ask, to what extent does that lead to brain aging? So Clotho is the Greek goddess of age, and a Japanese scientist about 10 years ago deleted a gene from a mouse strain and found that that mouse strain ages much more rapidly. He called it Clotho. He called the gene he deleted Clotho because of the relationship to age. And he sent us brains from his mice, and what we could compare is that what we found out is that brain aging alters both Clotho and cholinergic related microRNA levels. This is a paper under review in a leading journal. And the included microRNAs referred to cholinergic picture because microRNA 335, for example, targets all of the muscarinic receptors. So this one doesn't go down, but 150 declines with aging. So we do know about microRNAs. Now what about the correlation of that to Alzheimer models? So we do have RNA from mice with Alzheimer mutations like APP and tau. We could compare that to age. There is a comparison. So you could say that in some aspects Alzheimer's disease is accelerated aging. And when you look at transfer RNA fragments, those change as well, which was new but perhaps less surprising, and brought us to the question, so what happens in the human brain and cerebrospinal fluid under aging? And that's a study that was done by Shani Vaknin and Nimrod Madrer in the lab, together again with Ido Paldor. And when we look at cerebrospinal fluid, you see that different types of transfer RNA fragments go down with age. And they go down with age both if they come from nuclear origin or from the mitochondrial origin. But the mitochondrial levels are much more limited to begin with. In any case, anyone with mitochondrial disease would suffer, again, accelerated symptoms because of that. So we published that. We found also that in Parkinson's brains we see different profiles. And then we decided to focus on what's going on in the Parkinson's brain. So one day Nimrod comes to me and says, I found a consensus motif. I said, Nimrod, what do you mean you found a consensus motif? And the guy says, what do you think I've done in 8200 for 3 years? I was watching computer screens looking for consensus motifs. By the way, that's where he is now, since October 7th. Probably watching at screens looking for consensus motifs. But then we decided to look for consensus motifs in the transcription meaning. And what we found was an interesting paper by Igor Ulitsky from the Weizmann Institute, who said that long non-coding RNAs often include consensus motifs that are persistent with evolution even when the long non-coding RNA that inhabits them is changing. So the consensus motif is stronger than the host gene. What Nimrod found was a consensus motif of tRNA fragments. And when we look at that, we find that nuclear originated transfer RNA fragments from Parkinson's disease brains accumulate, while mitochondrial ones decline in Parkinson patients' CSF. And we do know that mitochondrial mutations accelerate Parkinson's disease. What you can see in this complex slide is that the PD genes are altered in a very different rate compared to, in the nuclear compared to the mitochondria. The mitochondria go down. In the nuclear originated transfer RNA fragments, Nimrod finds a dramatic increase of specific transfer RNA fragment subsets. We could prove that in brains of patients and in the CSF. And when we looked together, we can see the substantia nigra Parkinson's-related motif sequences go up in the brain, in data sets so far. Now we got from the NIH the real brain, so we need to prove that. The mitochondrial ones go down, and the ratio between them, because of that, is going up. And if we look at the space where these sequences accumulate, we see an increase with disease progression. So we asked the question, what about diagnosis? There was a paper published recently on early diagnosis of Parkinson's disease. And typical to the US medicine system, this was extraction of cerebrospinal fluid, which you would say, okay, you need a neurosurgeon to even start this analysis. Then laborious purification of a very rare protein, which requires a lot of lab work. And altogether you find an analysis that would cost about $6,000 per sample minimum, which is again typical for a medical system that is geared to improve treatment of the top millionaires class in a country without public medicine. What we did was to say, can we compare the validity of testing for this repetitive motif in patients that have early Parkinsonism? And what we find is for white and Hispanic populations, if you look at the bottom left part, we find that the ROC curves, the diagnostic prediction, is far better when you look for these sequences in simple blood samples. No CSF needs to be extracted. And if you seek association with the clinical prediction, this is much more reliable, especially when you divide the Parkinson-related nuclear originated to the mitochondrial ones, because mitochondria are damaged in Parkinson's disease, which is known for many years. So we are still working on improving that, which became difficult because Nimrod was recruited on October 7th, but we are trying to calibrate and improve the blood test by using qPCR primers. And last but not least, so how come? What do these motif sequences do? And apparently they can interact with ribosomal RNA, they can block translation, and actually we found a beautiful paper in yeast that was published 10 years ago by two Polish ladies that found in yeast cells motif-carrying fragments. They didn't know that they include this repetitive motif, but we look for it anywhere, I suppose. And they say this particular sequence blocks translation. So we have here interaction with two type TRFs. The motif-carrying TRFs interact with a translation-promoting TRF, blocking its activity, and at the same time, they may interact with the ribosome, blocking its activity. So we have a dual lock system that, again, we would like to complete improving. And the last but not least question, so what happens in treated patients? So treatment of Parkinson's disease starts in India 4,000 years ago and proceeded in the last century with dopamine replacement and deep brain stimulation. So what we did was to look for deep brain stimulation related changes. And what we find is that this treatment apparently reduces the repetitive motif sequence, and that in neuroblastoma cells we can see a parallel change when we attribute electrical stimulation. So electrical stimulation apparently reduces this repetitive motif, and because DBS is now used to treat other diseases, we would probably need to extend it to look at schizophrenia and depression and those other diseases that are subject now to experimental deep brain stimulation. So that was a rapid march through the issue that I tried to convince you that acetylcholine signaling is under consistent surveillance of small non-coding RNAs, including not only microRNAs but also fragments of transfer RNA, that cortical cholinergic pathways in men and women show distinct transcriptomic reactions to neurodegenerative states, that rapid stroke-induced changes in cholinergic gene targeting transfer RNA fragments replace microRNAs in the post-stroke patients, which would change their immune reaction and hopefully protect them from pneumonia. That mitochondrial TRFs in the brain from women with Alzheimer's disease show expedite cognition-related declines. Actually, this is the second paper in my life that was accepted without comments. And Jonathan tells me that this is bad because reviewed papers are always better than previously or immediately submitted ones. But it was nice for the student to got a job to go to thanks to that. That cerebrospinal fluid and blood transfer RNA fragments of either nuclear or mitochondrial origins show age and sex-related changes, also in Parkinson's disease, and that a consensus DBS suppressible TRF motif accumulates in the Parkinson's CSF and blood. And I think that I need to thank all the patients that contributed samples, not only to us, but also to the NIH and to the Rush collection and to all of the sources from where we got data. I feel that it's very important for me personally to proceed with comparing men to women differences in treatment. The transfer RNA fragments are a beautiful family to focus on, and we are getting there. And age-related issues are very important, as my grandchildren keep mentioning. And I'd like to finish by citing Marie Curie that says that nothing in life is to be feared. I'm not sure, but it's nice to read. It is only to be understood. Now is the time to understand more so that we may fear less. Let's hope that science will help us all to fear less. And thank you for listening. Yeah. Here's an expert question.
I was wondering about the difference between the tRNA fragments from the mitochondria versus the nucleus.
Yeah.
If you think, could this be actually a sign of some degradation or changes in the mitochondria, or the tRNA fragments are actually part of the mechanism of dysfunction?
Both, probably.
How do you distinguish between?
We do know which fragment came from what gene. They are not exactly the same, and we are starting to look also to what they can do. MicroRNAs the only function known for them is interaction with RNA targets. But TRFs are longer, up to 40 or even 50 nucleotides in some cases, and they bind proteins too. So we are now performing pull-down strategies to find which TRF binds what proteins, but we don't have solid findings yet. Yeah, Ran.
So there's a lot of interest in recent years in cell-free DNA diagnostics.
Mm-hmm.
And
yeah.
So the question is if what you're seeing in tRNA is basically the other side of the coin. Is it?
Probably.
So, which means that it's just a manifestation of the death of the neurons, right? That shed their tRNAs into the blood rather than something else.
I'm not sure, or else it might also reflect reaction of blood cells to the change in the brain.
This is the question. So
I don't know. It's extremely difficult to differentiate between the two. In microRNAs, the amounts that people find in the blood are far lower than those in the brain. So my assumption is that the second option is more likely to occur. But I don't know for sure because fragments are fragments.
But do you look in the PBMCs or in the plasma?
We do both. And we now, after fighting the shipper for 2 months, we now got samples from the NIH collection to enable us to do the tests in the CSF in the lab rather than using data sets.
do you have tRNAs that have fragments that are specific to cholinergic neurons? Because then you'll be able to see whether these are highly represented.
Yeah. So far, there are fragments that are particular to neurons, that I showed you. There are individual fragments that are particular to subtypes of neurons. We don't know about the ones we found yet, but their levels are much, much higher in the CSF than in the blood. That we know. And it's very difficult to run experiments and correspond by WhatsApp messages to the 8200 base where Nimrod is serving. So I hope that he will return soon and the work will proceed as planned. You're right, it's a type of liquid biopsy. Yeah.
Another question about your last results with the Parkinson's and the DBS. It wasn't clear to me if the changes are a response to the treatment, or do you see them also, or like there are Parkinson's related changes, right?
There are Parkinson's related changes. They do not react to L-DOPA treatment, that's for sure. In the DBS, we assume that that is in response to the treatment. And if you look at cell cultures in neuroblastoma cells, you see a parallel reaction. When you activate the cells, you see a reduction in the transfer RNA fragments that associate with ribosomes, which is the other part of the same picture. So yeah, it probably is. And because this is a treatment that is used today or tried today in many different diseases, it's of much wider interest. But we only looked so far, and this is a 3 years project, I think, that a lot of the lab team contributes to, and we're still not at the end. So it's tough to prove. It's very new, so yeah. Okay, so thank you again. סליחה?
מה תעשי כדי לעזור לנשים להיות יותר כמו גברים בארץ?
זו שאלה מצוינת, מפני שכבר שנים מדברים על טיפולים מיטוכונדריאליים ואף אחד לא הגיע לשום דבר קונקרטי. אבל אפילו להבין מה קורה, זה כבר חשוב.
להבין כדי לעשות משהו.
בוודאי.
אבל עכשיו שאת מבינה למה את לא עושה.
בחייך, אתה רואה מה זה? אנחנו נלחמים לגמור מאמר שאז יש לי שליש מהסטודנטים במעבדה כעת ואחת שנשארה עכשיו גויסה ליחידה לנפגעים. המנגנון הוא כנראה בלימה של תרגום.
אבל זה יקרום להרבה חלבונים לא להתעצר. למה דווקא הפולינרגים?
זה יכול לשפר את האימבלנס הקולינרג'י דופמינרג'י.
זה לא. תוקעת את הריבוזום, אז כל החלבונים.
נכון. ואני לא יודעת באיזה תאים זה עובד. התאים יכולים להיות חולשים. אנשים מאוד לא אוהבים.
יש לנו אבל סיפור עכשיו שטיפולים, מניפולציה, לא אמורה להיות עובדת, רואים את השמיני התנהגות מאוד חזקים, שונים, והדברים שונים בנפקות,
קיום. אם אתה רוצה לעשות סיקוונסינג, תתקשר.
מדבר עם חשבתי על התאים החולינרגיים.
אני אשמח.
תקרא את הדברים השונים.
מצוין. בכיף. אוקיי. תודה רבה, אחד שבאמת מומחה. בחייך. לא לקחתי את הדיסק-און-קי.